Let's start with the molecule of life, the DNA. First, we'll clear our basics, then, we'll move on to really dig up into the concepts of genetics. So, starting from the building block of DNA, a nucleotide. Basically, what a nucleotide is made up of? A nucleotide is made up of a sugar, a base, and a phosphate. This base is basically a nitrogenous base. Let's explore each component one by one, starting from sugar. Sugar is a vague term. This is carbohydrate, specifically a cyclic carbohydrate, and the cyclic ring which is found in nucleic acid is a pentose ring. Pentose is a 5 carbon ring. But there's a slight difference in DNA sugar and RNA sugar. DNA sugar has no oxygen at 2' position, but RNA sugar has. Let's dig up into the structure of a sugar. So we have got a 5-member ring, but the 5th carbon of the ribose is present outside of the ring, and the 5th position is occupied by oxygen. Each carbon of a pentose ring has got an oxygen and a hydrogen. Now let us try to understand what we mean by 2', 3', and a 5'. So the first carbon of a pentose ring is referred as 1' carbon, and the second carbon is referred as 2' carbon, and so on and so forth. So this terminology becomes quite important while referring to the replication, transcription, and so on, because we have got a 5' and a 3' and this all just refer to the position of a carbon in a pentose sugar. So now let's see what is the difference between a ribose and a deoxyribose sugar. So we can see that at a 2' carbon, if we remove an oxygen, what we have got is only hydrogen in it, and that, my friend, is a deoxyribose sugar. And please understand the meaning of the 1' carbon, 2' carbon, 3' carbon, and so on. That will help a lot while understanding the structure of DNA, and that is genetics basic 101.